Okay, I want to do a video on the mitosis meiosis worksheet. Granted, I'm trying to draw digitally. This might not be amazing, but I want to do the best job I can here to try to make sure you're on the right track, because this is usually a challenge for students, this worksheet. Students often think they know about mitosis and meiosis until they're told to draw something. So your worksheet showed you that there's 4 different chromosomes in this organism. And the first major part of the worksheet says to draw the 4 chromosomes after they have replicated and as they would appear on the equatorial plate in metaphase of mitosis, and keep track of correct shading and correct allele designations. So here's the metaphase plate. There it is. Okay. Now you have these 4 different chromosomes on your diagram, on your worksheet. Now you have to remember that's what they look like in, let's say, like G1 phase of the cell cycle. What we need to do is show them after they've replicated and now they're laying on the metaphase plate. Now what that's going to look like, let me just draw one of them here. So that first chromosome, the first white one, which I think white is maternal, is replicated. So let's first draw the centromere. I'll draw it right here. Here's the centromere. So what we have here are 2 sister chromatids stuck together. And we have gene B1 right here. And we have another copy of it on this one right here. And we have gene, I think it's R1. I can't quite see it on my screen. So R1 there and R1 there. It's laying on the metaphase plate. So this is a chromosome that duplicated and is on the metaphase plate. Also, you're going to have the paternal chromosome duplicated. And that one has B2. And I believe R2. And of course we can shade this chromosome to make it match the one in the book or in the worksheet. Okay. Then of course we have the smaller chromosomes. They're replicated as well. So there's one on each side of that metaphase plate. There's a centromere, and we have gene D1, and then the other one. And that was gene D2. Okay, so that's what you needed to have drawn for that one. Each of these chromosomes is duplicated. So we went from this to this, right? And so these are laying on the metaphase, plate. Let's draw it this way, like this. So that's what we're seeing there. And there's a centromere, which is what these black squares are. All right. The next question on the worksheet says, as you did for above in mitosis, draw the same 4 chromosomes as they would appear after they have replicated in S phase and are now on the metaphase 1 plate. There are 2 possible ways that the chromosomes can be aligned due to independent assortment. Alternative 1, both of dad's chromosomes can be on one side of the cell and both of mom's can be on the other. Alternative 2, one of dad's and one of mom's can be on either side of the cell or vice versa. Draw both alternatives in the space provided. All right, so how do we do that? Let's draw our metaphase plate. Again, on one case, both of moms are on the same side. So in this case, you're going to have both of mom's chromosomes have duplicated. Okay, just like before. Let me make sure I get the shading right. Let me bring up what the chromosomes are supposed to look like so I don't forget. All right, so here is gene B1. I'm just going to draw it on the one side now, but there's 2 of them, one here, one here. And we have gene R1. Down here, paired up with this chromosome, is the paternal chromosome. So this is gene B2 and gene R2, and this chromosome is the paternal chromosome. So these are paired up in a tetrad. Homologous chromosomes are paired up on the metaphase plate. That's what we're seeing there. And then we have over here the same deal. We have all the moms on one side. I'm gonna draw, well, let me draw this one shorter so it looks more like your worksheet. We'll put the centromere there, and we have gene D1, and then paired up with that one is this one. We have gene D2 on that one. And of course, if we wanted to draw the spindle is going to be attached to these. No, so I'm going to draw this one stubbier down here, like that, right? So there's our spindle. This is our centrosome, and our spindle attached. The other way to draw this, thanks to independent assortment, I'm just going to draw it smaller over here. This is 1 and 2. Let me just change colors just to make sure they stand out. So this one might be the same here. B1, R1. So this is one sister chromatid, there's another sister chromatid, right? And this one might be like it was on the other drawing. B2 and R2. But over here now, actually I didn't shade this one. Let me shade this one over here. Okay. Over here now, we might have dad's chromosome up here. Okay, so here, once again, let me draw that one into. So D1 is down here, mom's chromosome is down here, dad's is up here. So this is independent assortment. For each chromosome, it's a 50-50 chance whether mom's is on top or dad's is on top. Okay, and that's 50-50 chance for each and every chromosome. So those are the two ways that that could have lined up. Then it says, Part B Meiosis. As you did above, draw the same 4 chromosomes as they would appear after they've replicated in S phase. Oh wait, I'm sorry. I'm reading the same instructions. Let me go down. Part C. In the space below, consider only chromosome pair 1 and meiosis 1, so we won't worry about the other one. Show a tetrad and a single crossover event between non-sister chromatids with the crossing over occurring exactly in the middle of the space between R and B genes. Okay, and then we're going to show what would result from that. Okay, so now let me show you how to do that. Okay, so here we're going to draw a crossing over event. So I'm going to do this in diagrammatic form again, just with squares. So let's say we have mom's replicated chromosome here, mom's duplicated chromosome, and dad's, and they are connected. If we were drawing, drawing this really well, they would be connected, but I'm going to leave space between them so we can see what's going on. We'll draw their centromeres. We'll put them here. Even if I put them somewhere else on the other diagram, I don't remember. I'm going to put them right here on this one. Okay. There's our centromeres. We'll make this one mom, so this is R1. Oops, I don't know what I'm doing. Okay, R1 and B1 is right here, just like before. Okay, and it says that the crossing over event is happening right between the B and the R gene. So this is going to happen between sister chromatids that are touching in this case. So this crossing over event could occur right here. So let's draw this. So this one right here is going to be R2, and this one right here is going to be B2 on the dad's chromosome here, right? But these two are going to have crossing over occur. So this is going to be B2 still. This is going to be it's going to be B1 still. But right between these two, between B and R, crossing over has occurred. So now this gene down here, this one right here is going to have R1, and this one is going to have R2, and this part is now striped, and this part is now white. So this chromosome broke right here and flopped here, and this piece broke and flopped down here. And now we have this chromatid here and this one here are recombinant chromosomes. And it says what would happen if these were now going into different cells, and it shows 4 boxes. Where would these all end up? How would they be different? So this one's going to go this way, this one's going to go into here, this one's going to go into here, and that one's going to go into the one down there. So one of these cells is going to get this one, which was R1 and B1. This one's going to get a recombinant chromosome here that's got some of dad's stuff and some of mom's stuff on it. It's going to have, this part's dad's up here, it's going to have an R2 and a B1 allele on it. This one down here is going to have an R1 and a B2 allele on it. And this one down here is going to be just like the original dad one here. It's going to have an R2 and a B2. Okay, so these two cells here, this one and this one, got a recombined chromosome. It has some of mom's genes and some of dad's genes on it. So this is what happens when you go to make a gamete. Your gamete has some of your mom's DNA in it and some of your dad's DNA in it. And it's thanks to crossing over. That's where some of that variation comes from, but also independent assortment gives us variation. And remember, in a human cell, just due to independent assortment alone, there's 2 to the 23rd possibilities in terms of different gametes you can make. And then when you add in this, adding crossing over, the number goes way, 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 way up at that point. Okay, so I know this came out a little crude, perhaps, but I hope that makes some sense.